So today we're going to do a very simplified discussion on DNA and DNA replication, and specifically why DNA is the preferred nucleic acid for the storage of genetic information. So what is DNA anyway? In the most basic sense, DNA, or deoxyribonucleic acid, is a double helical molecule that contains the genetic information of cells. The information contained in DNA is what ultimately will determine the expression of genes and alleles in an organism. Now we will take a look at the specific structure of DNA. DNA is composed of two main parts. The most familiar parts are probably the nitrogenous bases, which are divided into purines and pyrimidines. The purines, adenine and guanine, pair with the pyrimidines, thymine and cytosine, respectively, to form the rungs of the helical ladder of DNA. They are linked together via hydrogen bonding. And you can see those here, where adenine is binding with thymine and guanine with cytosine. And those hydrogen bonds are occurring here, indicated by the dashes. The other main parts of DNA include the the other main part of DNA, is the phosphate deoxyribose backbone. And some key features of the backbone include that the phosphate groups, as you can see here, are what give DNA its acidic qualities, and the deoxyribose sugars, as you can see here, are vary from ribose sugars, which we will discuss those further and later on in the presentation and why that's significant. Another important feature to note is that DNA is antiparallel, and this is in respect to the numbered carbons on the deoxyribose sugar. One side of the helical ladder proceeds from the 5' to 3' end, while the other side goes 3' to 5'. Okay, so now we'll do a very, very brief overview of what RNA is. And RNA, or ribonucleic acid, varies from deoxyribonucleic acid on two main fronts. First is that the nitrogenous base thymine is replaced with uracil, and secondly that the hydroxyl group is removed from the 2' carbon in DNA. So you can see that here, where RNA has the hydroxyl group while DNA does not. So why is this significant? These differences in RNA and DNA can explain why DNA is favored over RNA. Because the hydroxyl group is removed from DNA, it makes it less susceptible to hydrolysis, ultimately making it more stable, which, having a more stable DNA molecule is important because it's going to actually store and hold on to genetic information, whereas RNA is more of a messenger molecule and will ultimately be degraded over time. So now that we've looked at the basic structures of RNA and DNA and why DNA is the preferred molecule to store genetic information, we will explore DNA replication. DNA replication requires several molecules to begin. It requires a template DNA strand with which to replicate, a primer to initiate the starting frame of replication, deoxynucleoside 5' triphosphates to help build and generate two new DNA strands, and the key players, enzymes, to not only add nucleotides to the new strands, but to carry out a number of other reactions important to DNA replication. So here we have a depiction of the replication fork where DNA replication is occurring. We're beginning here, or actually we begin over here, but this part of the DNA is still tightly supercoiled. So in order for replication to begin in this area here, we have to unwind the DNA, and topoisomerase is the enzyme that helps unwind DNA. From there, helicase then begins to break apart the hydrogen bonds between the nitrogenous bases, kind of like unzipping a zipper. This ultimately splits DNA into two separate single strands, and these two strands are defined as either the leading strand or the lagging strand, depending on the anti-parallel orientation. The leading strand, as you can see down here, is where the template strand runs from 3' to 5', can simply have an RNA primer added to it, and then DNA polymerase can begin to add nucleotides in the 5' to 3' direction. This is different, however, for the lagging strand up here, because its template strand runs in the 5' to 3' direction. So that would ultimately have the new strand running in the 3' to 5' direction. And there are extra steps to this because DNA polymerase can only add nucleotides in the 5' to 3' direction. And the way biology works around this issue is that nucleotides can be added in the 5' to 3' direction in short fragments known as Okazaki fragments, as you can see right here. These tiny fragments of DNA are then covalently linked together using the enzyme DNA ligase. And ultimately, this will create 2 replicated strands of DNA. This concludes my presentation. Here are my photo credits and references if you're interested. Thank you very much.